if we couldn't build a system that mimicked what the brain does, then we don't understand what the brain does.
How do you think AI might help answer a few of those big questions?
The things that we consider hard and challenging turn out to be easy. They're amazing in language, but they still can't do the things that all animals can do. And I think that sort of trying to understand why that is will tell us a lot about how our brains work and what the sort of real challenges are. And that's actually what a lot of my current research is on. It's trying to understand why are those things hard? Why is vision hard? If you think about the most commercially important application of vision decisioning systems is self-driving cars. It's incredibly impressive what vision systems can do. They're still sort of not doing it the way we do it. And I think that means that they're ultimately going to keep failing on edge cases. There's a fundamental problem. It's going to be solved by building systems that not only solve the problems we do, but solve them in the same way that we do. And the way to get them to solve them the same way is that their foundation should be similar to the foundations that we humans had as we evolved. We could train these systems potentially with orders of magnitude less data if their foundations were the right foundation.
You've spent a lot of time studying the connectome. Let's start there. What is a connectome?
The connectome is a term for basically the wiring diagram of a neural circuit. Connect as in connection, and ohm was inspired by all the omics from genome, transcriptome, where the idea is that you get a complete list of all the connections. What we do is a little bit different from what a lot of labs that are attempting to figure out the connectome do, in that we use a different technology. But the underlying goal is the same. It's the recognition that how a circuit is wired up potentially has a lot of impact on how it works and can provide insight into how it does what it does.
Yeah, it's a deeply fascinating and very ambitious, I think, field of neuroscience. What brought you into the field initially? How did you get interested in it?
Yeah, that's a funny story. It's my trajectory has taken a lot of pivots over the years. A lot of people would say that Connectomics is a fancy 21st century word for what people traditionally have called neuroanatomy. And ironically, I was an MD PhD student, and I actually failed neuroanatomy because it was
Hard subject.
well, it was a lot of memorization, and to my mind, it was the least fun. And I guess that in some sense is part of what drew me there, which was that I came to recognize that on the one hand, it's incredibly important to understand how things are wired up. But on the other, I sort of came to neuroscience with, if not a physicist full training, at least a physicist background, where the idea is that there should be simple principles. And my frustration came from the fact that as I was doing physiology and trying to understand the neural circuits underlying auditory behavior, auditory decision-making, there was a lot of work in the circuitry that I thought would be super useful to know. And what my dream was that you ought to be able to just sort of type in on a computer, is X connected to Y? Does this hypothesis for how the circuit is wired up, does that make sense? And it was typically not possible to do that, mostly because we just didn't have the sort of depth of information that we needed. And I guess I started developing a new technology to figure this out, very naively thinking that it would be relatively straightforward to use this technology to get a deeper understanding of the wiring diagram, and then we could go back to the original questions. And now, well over a decade later, we're still sort of working on the technology itself.
I never thought someone could thought that that would be relatively straightforward to map the most complex system that has ever been created in the known universe. I
Yeah.
don't know.
Yeah. I think naive would be the term that I would apply to myself. Um,
Optimistic, I know.
Incredibly naive. Optimistic. There we go. That's the term I probably should have used.
Yeah.
Yeah.
What are some of the things that we've learned about the human brain from the field of connectomics as a whole, from modeling simpler model organisms?
Yeah. So I should back up a little bit and say that I work exclusively in animal models, um, primarily in rats and mice. And on the one hand, it's pretty obvious that rats and mice are different from humans. But the reason I study them is that they're experimentally accessible. And because honestly, I think that the depth of our ignorance about how brains work is so great that if we could only understand rats and mice and perhaps even other simpler organisms, we would be very close to understanding how humans work. So, um, I obviously humans can do a great number of things that rats and mice can't. But on the other hand, um, we evolved fairly quickly from creatures who weren't that much more sophisticated from rats and mice. And I'll just throw out a little, I mean, maybe we'll talk about this more later, but the way I think about it is that the genome specified the connectome, and the difference between the genome of a chimp, our nearest relative, and of a human would fit on an old school floppy disk. It would be about 1 million little pieces of DNA, characters of DNA. And the difference between a rat and a human is about 10 times that. And that's a tiny fraction of everything in our genome that makes us up. So that's sort of, for me, part of the inspiration for thinking that, or the justification for thinking that if we could understand how rats and mice are wired up and how their wiring gives rise to their behaviors, we would be a lot closer to understanding the really fundamental things that we don't understand about humans.
Yeah, absolutely. And then to be more specific, what do you think are some of those findings that we have discovered about the human brain from the earlier work in connectomics, from modeling, I think first the C. elegans nematode, then the Drosophila melanogaster. I think there's multiple connectomics projects on the full rat and mice brain now. I'm sure you'll have a lot more information on that. Have we discovered anything fundamental other than that's a really hard thing to model brains, to model nervous systems?
Yeah, so um, I think what you might be looking for is a simple one sentence smoking gun kind of discovery, and that's actually what we don't have, right?
Yeah.
The way that this kind of discovery works is that you end up investing years and decades, dozens, hundreds of labs work together, or, work as a field to establish like new fundamental principles. And then at some point, they sort of percolate out through a breakthrough. So if I'm going to be honest, I'll say that the technologies that we can use for the human brain, which, for obvious reasons are different from the kinds of technologies you can use for rats and mice, they're kind of limited. They're fMRI for the most part, which allow us to establish functional connectivity. And we could. I'm not really. That's not my field. I'm not an expert in that. But we have learned in the 2 or 3 decades since fMRI has been applied to the human brain. We've definitely made advances, but they're not sort of the kinds of home runs that I think we'll get in the future using the combination of these previous, of these newer techniques, and sort of the foundation that we've gotten there, and by studying humans. So, we have, for example, begun to understand what constitutes the normal functioning of the brain, and there are hints in terms of activity. I should be really clear that in humans, a lot of the work is not on the structural connectivity, which is what's physically connected to what, but rather the functional connectivity, which is what brain areas are active when other brain areas are active. And so some of that functional connectivity work has begun to bear fruit in understanding what's going on when we think about stuff, when we feel stuff, and also in pathological conditions, for neuropsychiatric conditions or neurodevelopmental conditions. But again,
Right.
the work that I'm doing is, on the one hand, it's laying the foundation for that. Um, it's the work that is going to, in maybe in the next decade, provide some of the breakthroughs that we'll need to understanding some of these conditions at a deeper level.
Yeah. Let's dive into your work. I'd love to. Can you give a little overview of the connectomics research that you've conducted and the quite novel approach that you've adopted, which I was again reading about and I think is really, really interesting and really fascinating. So I'll let you describe it in a much more succinct way than I could.
Yeah. So what got me there was I was studying the relationship between neural activity in rats and mice and how they make auditory decisions. I was interested in the circuitry by which they sort of take information from their ear, process it, goes into the cortex. They make decisions about, for example, whether a sound came from the right or from the left, whether it was a high frequency sound or a low frequency sound. We know a lot about how that information comes into the ear. And eventually it goes somewhere into the brain, into the cortex and into subcortical region. And then in some sense, it comes out the other side as a bunch of decisions. You have to move your muscles. In this case, we trained rats and mice to go to a reward port if they guessed right about where the sound was coming from or whether it was high or low frequency. So we know a lot about how information comes out of the spinal cord, how neural activity controls the muscles. But what happened to that information in between? How is it represented? How did the signal of a sound go through these different brain areas? And that's where we kind of got lost. And so my idea at some point was that it would get a lot easier to treat that information if we could know how all the neurons are connected to one another. And when I started that, the main approach for sort of understanding how single neurons send their information around was there were two approaches. One was based on optical microscopy, and what you do, it goes back over 100 years. You fill a single neuron in an animal with some kind of a dye, and then you wait for that dye to percolate through the axonal tree of that one neuron, and then you slice up the brain into parts, and you can track that one axon as it passes wherever it goes. The problem with that is that the rat brain has maybe 100 million neurons. The human brain has 100 billion neurons. If you're going to go one neuron at a time, things are going to move very slowly. Yeah. So there were people who were recognizing that one neuron at a time is slow. They figured out that you could fill neurons with different colors. But the problem is that the kinds of ways we're filling them with colors and the kinds of microscopes we have really only allowed us, although in principle we could fill two neurons with 200 different colors. In practice, we could only resolve 4 or 5 with microscopy. And so my idea was that instead of filling a neuron with a color dye, whether it's genetically encoded or not. We could fill it with a genetic barcode, a sequence of DNA, A, T, G, C, just a random sequence. And in principle, we could fill all neurons in the brain, or a good fraction of them, with these random sequences. And the great thing about random sequences is that there are a lot of them. If you count the number of possible random sequences of the 4 nucleotides ATGC, you get if you consider a random sequence of 1, there are 4 possible random sequences ATGC. If you consider random sequences of 2, there are 16, 4 times 4, because ATGC in the first position, ATGC in the second position. And if you consider a random sequence of 30, you get up to 10 to the 18, which is a really big number. So, and it's easy to make a random sequence of 30. So, so the idea was to fill these neurons with these random sequences, and then we could use the incredible advances in DNA sequencing that have sort of come around in the last 2 decades, and we could read out those sequences using DNA sequencing. And that has been the idea, and that's what we're pursuing, is basically we fill neurons with random sequences, and then we use DNA sequences to figure out what neuron is filled with what sequence. And the trick is that we figured out a way that each neuron sends its own unique random sequence all throughout its axonal tree. And then we can, figure out where a neuron is sending its axon throughout wherever in the brain it's sending its axon. I should add that there's an alternative method, which maybe we could talk about more, which is called serial electron microscopy. And there the idea is that you use electron microscopy to track every single axon and how it's connected to every single other neuron in some brick of tissue. And in principle, that's an awesome approach. And to date, it provides some of the most complete and reliable information about how brains are wired up. The challenge there is that you have to slice the tissue extremely thinly. And when you take a cross-section, and by extremely thinly, I mean, tens of nanometers. And if you take a cross-section of the tissue, you have to align what you see in one cross-section with what you see in the very next cross-section. And each time you sort of make this alignment, it's like taking these, you imagine sort of, cutting these pictures and then aligning these little round circles that are the axons or dendrites. And if you make a single mistake out of the tens of thousands of sections in assigning one little circle to another circle in the next one, then you basically make a mistake in reconstructing the entire axonal tree. And so it's incredibly challenging from both an experimental point of view to get reliable, beautiful pictures without making, the error rate has to be, 1 in 10,000, 1 in 100,000. If you're tracing an axon a long distance, it could be 1 in a million. Yeah. And then it takes really sophisticated software or a lot of patient people to reconstruct those circuits. And so, yeah.
I dug into this for a video a couple of months ago because I'm sure you're aware there was a really interesting paper that came out that basically was able to use electron microscopy to model 1 millimeter of brain tissue. It was over 1 million gigabytes, 1.4 million gigabytes, I think. One of the things I learned while researching for that video, which I found hilarious, was when they did the nematode modeling, as you said, they had to hand sketch those things that came out. So there was a team of scientists that spent 10 years basically stenciling into nematode brain. And that just, anytime anyone ever complains about work, I think that's a good thing to think about. You didn't spend 10 years basically penciling in sketches of nematode, of worm brain. But that whole process was phenomenal. They got a really, really high resolution, 1 millimeter cubic square of brain tissue, and they discovered loads of things about that. I think one of the really interesting things was that the in that sample there was a 2-to-1 ratio of glial cells to neurons. That's one of the things that caught my eye. Anyway, I'm curious to get your thoughts on what you think that means. So in this, just to reiterate so that the viewers, listeners understand, is that they got this 1 millimeter square model of brain tissue. And once they reconstructed all of the, I don't know how many, several thousands of basically images of electron microscopy they took, they used really advanced Google algorithms to piece them all together. And then they were able to estimate how many neurons, glial cells, oligodendrocytes, everything was in that sample. And there was twice as many glial cells as neurons. What do you take of that? And that was a pretty interesting finding for me.
Yeah, so just for your viewers, I'll just remind them that the traditional view is that the sort of interesting part of how brains work is that neurons send electrical signals to other neurons. Sometimes they send electrical signals to nearby neurons. Sometimes they send electrical signals to distant neurons. But when push comes to shove, it's important to remember that, as some people say, the brain is just meat, and it's not just an information processing device. And there have to be brains, the cells in the brain, the neurons, have to do all the things that cells in other parts of the body have to do. They need to get, if a lot of that volume is also taken up by blood vessels, right? Because neurons are very energetically demanding. And neurons are also sort of unusual in that they don't divide. They're what biologists say are terminally differentiated. And so you have to take really good care of the neurons, after development. I guess we continue to get a lot of extra neurons in our first few years of life. And then there are a couple parts of the brain where neurons continue to be born. But for the most part, most of the neurons in the adult brain are neurons that, have been around for decades. And once you lose it, you lose them. So we have to go to a lot of trouble. Biology goes to a lot of trouble to take care of those neurons. And some of the support cells, in fact, the main sort of group of support cells are these glial cells. And interestingly, these glial cells are often the ones that are implicated in disease processes. So, for example, in neurodegenerative diseases, in a lot of cases, we don't fully understand what's going on. And it's complex because biological systems are complex, but a lot of evidence suggests that in a lot of cases of Parkinson's or Alzheimer's, what's going awry are these glial cells. So these glial cells are the ones that are responsible for making sure that the neurons are actually able to do their job. They, their neurons communicate through synapses, which means leaving one neuron and going through another neuron. And they have to go, for example, through the extracellular space. And to keep that extracellular space clear and clean so that these signals can be transmitted sort of effectively without being contaminated by the junk in the extracellular space, you need glial cells. So I think part of what that's telling us is it's reminding us, what a tricky job it is to keep the neurons happy and healthy.
Yeah. So you don't believe, I think that there's some ideas out there. I think the evidence might be limited that glial cells might not be just support cells. They might actually play a fundamental role in information processing, even things like consciousness in a macro scale. Do you think that there's any evidence that glial cells, or what does your intuition say there? Is it still that neurons are the star player and glial are a support team? Yeah.
Yeah, I mean, obviously we don't know, and I could be wrong, but there's no question that glial cells respond to activity. And I'll just say that as a graduate or as a postdoc, I had a project where I used glial cells as a monitor of global neural activity. So it turns out that these glial cells are, among other things, taking up glutamate, which is the excitatory neurotransmitter, which is how neurons send information long distances. And when there's a lot of glutamate being released, glial cells take that glutamate up, and you can see it in their electrical activity. And when, if you fill them with a calcium indicator, which is a way of telling what's going on, you can definitely see the evidence. You can see what I would say are echoes of the neural activity in the other neuron. I think it's a little hard to define exactly what it would mean to be involved in a computation, but I guess to the extent that we could sort of agree on a definition, I'm still sort of in the old school view, which is that nope, the interesting parts of the sort of fast computation that are responsible for things like recognizing a face or a dog, reacting to an expression on your face, generating the string of words the string of muscle commands that I use to generate words. I think that happening through fast electrical communication, and the glial cells, have to keep up, and so they reflect that. And if you break them, then it won't work. But that, in some sense, the sort of to me, the challenging part, from a sort of computational point of view, are the messages that are sent via synapses between neurons.
Gotcha.
And so I guess I definitely, there are people who believe otherwise, and some of them are very passionate about that. But that I'll just say that in this particular way, I remain sort of conservative, mainstream scientist, and I'm not on board. I'm not on board that train at this point.
Gotcha. Well, that makes a lot of sense. And I think that's a very good synopsis on what the glial cell function is in the brain related to pathophysiology, the support cells, why they're so important, why they're critical, why the brain relies on them, why neurons require them. Let's get back to your own methods, because we kind of left it hanging there a little bit. So the current understanding is that you're inserting DNA sequences into these neurons. Is it only neurons, and what's the delivery vehicle?
So right now, the way we do it is we create a tube full of barcoded viruses. We use a particular kind of virus called SynBit for technical reasons, but in principle, it could be done with other viruses. So in neuroscience these days, it's not uncommon. In experimental neuroscience, it's not uncommon to inject viruses into the brain. That's one of the main tricks that we have. And it turns out each little viral particle is essentially a piece of DNA or RNA that's wrapped in a bit of protein, sometimes with a bit of fat inside. So it's basically a delivery vehicle for these kinds of materials. And all we do is we use some tricks from molecular biology so that each little viral particle contains a special protein that we've engineered. I'll tell you about that protein in a second. And it contains a unique sequence of DNA, a random sequence. So each of our little viral particles has a different sequence of DNA than every other viral particle. And so what's special about the protein that we've engineered is that it can grab a piece of RNA, an engineered piece of RNA. It's the one that we've engineered to carry this unique sequence. So this protein, which is modified from proteins that normally go out to synapses, this modified protein grabs the RNA barcode and drags it out to the synaptic terminal. And that was sort of the trick that we came up with to cause the barcodes to traffic to distant parts of the neuron where they would normally not go. That was sort of the main trick. Once they're out there, then we can then we know that there's a neuron that we inject the virus at. For example, I'll just say the auditory cortex, and we wait a few days for the virus to do its thing. And now we have the cell body, the soma, which is full of a barcode. So we have one neuron that's full of barcode 12, a second neuron that's full of barcode 42, a third neuron that's full of barcode 6,227,000, etc. And we have thousands or tens of thousands or hundreds of thousands of neurons, each of which has its own unique barcode, and that each of them sends its barcode out to wherever it sends its axons and dendrites. And then that's how we can figure out that, we're now looking at a part of the brain in a mouse that can be millimeters away, centimeters away. If we could do this in a giraffe, we could look at barcodes that are meters away, because we could put the barcode into the brain and look at the bottom of the spinal cord, which is meters away. We don't currently work on giraffes, but if we did, that's just to give you the idea that we're transporting these barcodes long distances. And that's our way of establishing that the barcode that the soma here, soma 12, has is sending its axon to this part of the brain, that part of the brain, some other part of the brain. And we can potentially even use that to figure out the synaptic connections between this neuron and some other neuron. Although I will say that that was the original ambition, and that is still a work in progress. That doesn't work as well as we would like it. But the part of just figuring out where the axons go, that has been working well and yielding fruit for quite a while now.
It's a really cool innovation. Yeah, it's a really cool innovation. That's what my next question would be, is so you're able to very well determine if neuron A connects up to neuron B, but what you might not be able to figure out is exactly how, how the dendrin.
So the word connection is used differently by in different subfields. A neuron sends out its axon to some distant place, and then once it's there, it makes synaptic connections to other neurons. When people talk about the human connectome, we're satisfied with just understanding which brain regions are connected to which other brain region, without knowing which single neuron is connected to what other single neuron. In C. elegans, it's all about which single neuron is connected to which other single neuron synaptically. And right now, that's what we would like to be able to do with this method. And currently what we can do is say that for 100,000 neurons, we can say that each of those 100,000 neurons, we get a list of the target to which they send their axons, and we can get that with sort of micron resolution, but we can't yet say that neuron 12 is connected to neuron 5277 and all its 10,000 partners. That's the work in progress that we would really like to be able to do. But even without that, even just by knowing who's sending their axons where, we can learn a lot about sort of the logic by which brain circuits are generated.
Yeah, it makes a lot of sense. Paint a picture for what the output of this looks like, because I think people can picture like electron microscope, you're just taking loads of photos, right? You get a model. What's the actual output of this DNA sequencing? Like, what are you able to create that you can actually look at?
Yeah, so there are really two versions of it. I'll start with sort of the early version. In the early version, the whole reason, just to back up a little bit, the whole reason that we were interested in converting the problem of wiring into a problem of DNA sequencing is that DNA sequencing over the last few decades, since the Human Genome Project, has gotten incredibly fast and efficient and cheap. So the Human Genome Project, which was figuring out all the nucleic acid in a single human, cost about $1 billion, right, for 3 billion nucleotides. starting in the mid 2000s with the rise of so-called short read sequencing and really dominated by the single company called Illumina, the price just plummeted. It plummeted faster than Moore's law, which is the rate at which computers get faster and cheaper. It's why, your cell phone has more compute power than a supercomputer from the 80s or 90s. But DNA sequencing got better and cheaper faster even than that rate. And so by the mid 2010s, the price of human genome, which is just, I keep mentioning that just because that's sort of the reference that people use, but the cost is per nucleotide. So the cost of a human genome sequencing went from about $1 billion to less than $1,000.
Yeah.
So, just a tremendous increase. And so when I started doing this, it was clear that prices were going to go down fast. So the first version of this method was to fill a bunch of neurons with barcodes, and then we wait a couple of days for the barcodes to go whatever, and then we dissected the brain into what we call little cublets. And in each cublet, we would just send the DNA in that cublet for sequencing. And so what we could tell is, this cublet, the source cublet, had barcodes 1 through 100,000, and each of these sort of target regions contained some subset of that. And so what we got back was just a bunch of sequences by cublet, and then we could do a little sort of data processing. And the end result is a big table that says neuron 1 projects to area A, C, D, F, and H. And neuron 2 doesn't go to A, it goes to B. And so that would be just that list. And we get that for, thousands or hundreds of thousands of neurons.
That's some Excel spreadsheet.
Yeah, it would take a very large Excel spreadsheet. Exactly. Actually, we do everything in MATLAB, but at least now, people are doing it in Python.
Yeah.
But so yeah, it's just, and from that the reason that was exciting to us was that in the old days, if you sort of painstakingly reconstructed a single neuron axonal arborization, you'd have one data point. But by getting, 10,000 or 100,000 in a given animal, we could start doing sort of statistics on them. We could see what the patterns were. So the other method relies on the fact that it turns out that when all is said and done, DNA sequencing is actually an optical method. And I didn't even sort of think that through when most people who get their Illumina sequencing results back don't really think too hard about what is going on in the machine. You hand the sequencing facility a tube, and they hand you back the spreadsheet, and you don't really worry about how that happened. But if you sort of break open the Illumina sequencing machine, it's actually all optical. And so we realized that instead of cutting the brain into bits and extracting the DNA and sending it to the sequencing facility, we could do the sequencing in situ. We could take a brain slice, and then we could actually do the sequencing while the neurons and the axons and dendrites were still intact. Not alive, but intact. Yeah. And so at that point, what we get, the raw data, is a sequence of images where each neuron gets a color based on ATGC, right? So each of the nucleotides ATGC is one color, but then we go back, we look at the next nucleotide in a sequence, and each neuron, each axon, each dendrite potentially gets a different color. And by aligning those those slices, we can reconstruct for each point in space, for each axon, for each dendrite, for each soma, exactly what barcodes it was expressing. Gotcha. And so that basically we're doing in situ sequencing, and the end result, if we combine that, instead of being a spreadsheet, it's a picture of the the barcode expression, the sequence, the barcode sequence in each neuron in the brain, at least of the ones of the neurons that we have infected with barcode. So the way I think about it is, I told you earlier that there was this approach for filling each neuron painstakingly, and then the next generation called Brainbow, where you filled each neuron with a palette of colors. That technique, Brainbow, where you fill each neuron with a distinct color, was the inspiration for this technique, and we've sort of overcome the limitation of that, which is that a given microscope can only resolve 4 colors. So instead of trying to resolve 200 colors at once, we resolve 4 colors, and then we strip off that color, and then we resolve which of 4 colors that same neuron expresses, and we strip that off, and we do that, 30 times, and we have 30 nucleotides. So it really is returning to the original inspiration for this approach, and you now get a picture of where all the neurons that you've infected send their axons and dendrites. We call that approach.
Sparse, yeah, so cool. And what have you found when doing this? Has anything came up where you're like, well, that I wasn't expecting that? Is there anything surprising that you've learned?
Yeah. So one thing that there are a lot of, I guess there are 2 kinds of answers to that. So, by doing high throughput sequencing or high throughput connectomics, you can sort of figure out the patterns that might have been difficult to figure out early on, or without these kinds of high throughput methods. And I'll tell you the kinds of things that I find. So we collaborate with a lot of people. We also have a facility where people just send their samples and then they answer their own scientific questions. And, there at this point, there have been dozens of papers published using this technique, and each one tells its story. And, the exciting thing is, in some sense, that instead of being the whole story, this is part of a larger story. And each one of those larger stories you could fill an interesting podcast with. I'll tell you what I have been excited about using it for, which is I've become really interested in the question of how you wire up a brain in general. And it's a very challenging problem, because if you think about the number of connections in a human brain or even a mouse brain, it's astronomically large. The number of connections is just astronomically large. And even though we have a lot of information in our genome, there's not enough information for the genome to specify for each neuron who its partners are.
Right.
And the what that tells us is something that developmental neuroscientists already knew, which is that you don't get a list, you don't get a wiring diagram, you get a list of instructions for how to build a brain. And over the last few decades, we've learned a lot about the molecules involved, the mechanisms involved in wiring up a brain. But in some sense, it's not clear to me that we really understand the principles. And one of the principles that I've gotten pretty excited about is that the rules that specify the brain are often pretty random. They're coin flips. They involve a lot of coin flips. The rules says something like, hey, if you're a neuron of a particular type, send your axon to this region and connect to, a third of the neurons of some other type. It doesn't say which third. It says, about a third.
Stochastic.
What's that?
So it's stochastic.
It's stochastic, exactly. And so one of our earlier findings, in fact, the very first sort of significant publication using MapSeq, was that so we looked specifically at a circuit in the visual cortex, and we asked from the primary visual cortex to secondary visual cortex, how do neurons send their axon? And the first thing we learned was that a given neuron, so there are about 7 so-called higher order visual areas that we looked at. And what we learned was that, first of all, that a given neuron sends its axon to more than one of those, right? So it doesn't just send it to area LM, for example. It sends to LM and typically another one. And so you might think, okay, well, then maybe it just picks 2 or 3 randomly. And the answer is not quite. What it does is that it picks subsets of them relative with higher, in some cases, it picks subsets with higher probability than other subsets. So it doesn't just sort of wire up one at a time. It doesn't wire up to any of the 7. It will tend to wire up to areas 2 and 4, and another neuron will be more likely to go 3 and 7, and maybe 6. And so the rules, although they're random, they have sort of complicated structure. And so then what we did was we scaled that up not just to the wiring of the visual cortex, but of the entire cortex. And what we expected was that there would be a lot of these complicated rules where, okay, if you're going to send your axon here, then you don't send it there as well. And to our surprise, what we found was that at the level of the whole cortex, this idea that if you simply know that the visual cortex sends its axons with low probability to this area, somewhat higher probability to this area, and even higher probability to this area, then knowing that at sort of the regional level is enough to tell you that what if each single neuron will do. And in other words, the rules for long distance wiring are really simple. It's just a list of, it appears to be just a list of, here are the long range targets. Now, go, if you're a given neuron, wire up to 2 or 3 of them at random, and that's enough. And that was really surprising to us because we expected that it would be more like the visual cortex, or there would be more structure. Which I think what's going on is that for your highest probability target, for the ones that are most important, you go to a lot of sort of trouble to fine tune it, or possibly that fine tuning happens through activity. But to first order, the way one cortical area figures out where to connect to other cortical areas is just a bunch of coin flips. And I'll say that one interesting implication of this, if I could just talk a little bit more about this.
Absolutely.
It's not just from my work, but integrating from a lot of other work, is that we're used to thinking about there being a tension between genes and environment. That's usually how the question is posed. Nature or nurture, genes or environment. But it's always been a little troubling to me because if you think about identical twins in humans, right? They're similar in a lot of ways, but they're still different people. They have the same genes. They have really similar environments if they're raised in the same household. So why are they different? And the usual answer, and the one that I sort of went with, was, well, we think their environments are the same, but, they're not quite the same. And maybe tiny differences in environment lead to large changes in, outcome. And I was sort of vaguely uneasy with that, but I was like, okay, maybe that's what's going on. But this raises the possibility that there's a third component that until recently we've been overlooking. I'll say that I first really had my eyes open to this by Kevin Mitchell's book from
It's a great one. Innate.
Innate, yeah, exactly.
Yeah.
Wonderful book.
Yeah.
Brilliant book. And he really is the one who drove this home, and I think this is really consistent with that, is that there's this third component, which is randomness, coin flipping. When you're wiring up the brain, right, these connections, although what the genome has specified is, what the probabilities are, each individual brain is going to have its own unique wiring, and that that unique wiring is going to be what, in many cases will determine the differences in who we are and how our brains work. And it explains, for example, something that I think has always been sort of tricky to understand, which is there are conditions like schizophrenia or autism where concordance between identical twins can be as high as 50%. So if you have identical twins with a particular genetic lesion that is likely to cause autism or schizophrenia, if one has, just if you go across identical twins, if one identical twin has schizophrenia, there's a 50% chance that the other one has it. One has autism, there's a 50% chance that the other has it. That's much higher than fraternal twins, which I think is closer to 5% or something like that. So the genes, but it's not 100%. So why is it not 100%? I think this sort of explains or provides a model for thinking about why it's 50% and not 100%. That there's something in the genetic makeup that predisposes the circuit to produce, something like schizophrenia or autism, but it's not with 100% certainty. There were some coin flips early on in development, and if it goes one way, then you end up with one of these conditions, and if it goes another way, you don't.
Right.
I guess in some sense, that's one of the reasons that I'm finding that's really interesting, is that this understanding that getting the statistics on brain wiring that would help us to understand what is that extra component beyond nature and nurture. This third sort of role in determining, in nature determining who we are.
Right. That was a really good synopsis again. And we're huge fans of Dr. Kevin Mitchell on this podcast. He was my past neurogenetics professor, so I got a great education on neurogenetics from him, and he was the first guest on the podcast. He's a
brilliant speaker and absolutely, brilliant writer.
And a nice man as well. What do you think that stochastic nature of how the brain develops tells us about the broad variety of pathophysiologies that can also develop? Is that inherent randomness somehow built into the very broad spectrum of different atypical development patterns that we see? Like autism, we have one label for, but now we're seeing is probably hundreds of things of just atypical behavioral manifestations. Do you think it's the stochastic inherent randomness underneath that is kind of underpinning those atypical syndromes that we have labels for, but are nowhere near as discrete as those labels might indicate?
I think that's exactly right. I think that what your genes specify, and it's not just pathophysiology. It's sort of an arbitrary label, what we call pathophysiology.
Absolutely.
I mean, I'm just reminding there's a huge range of sort of human and of course non-human behavior, and that's actually a positive thing. We don't want everybody behaving the same way, not just like in a moral sense, but in an evolutionary sense. You need a whole range of sort of behavioral strategies, and it's helpful. For example, back when we were a small tribe, it was helpful to have some people who are timid, some people who are bold, some people who are I'm a night person, so we're always being told that you should early to bed and early to rise. In our evolutionary history, my ancestors were the ones who were awake at night and noticed that the bear was about to come to camp. I perk up at night, so I feel like my ancestors were the ones who were protecting the camp at night.
Well said.
I think having what's it?
Well said. The variance is essential to the human race in every way.
And so given what our society is like now, there are certain of these behavioral phenotypes that are perhaps not as easily accepted, right? But they're all part of this spectrum. So yeah, I think it's absolutely part of it. Now, of course, there are things that I think we can all agree are outright pathophysiological. And I think in some of those cases, early neurodegeneration, right? Clearly problematic. We don't really, of course, understand all the causes of that, but we know that, not only are these conditions typically multigenic, right? They require the right combination of genes, but even as I say for identical twins, even in those cases, they have incomplete penetrance, meaning that there are some coin flips. And I think those coin flips, a lot of them are the ones involved in wiring up the brain. But this isn't just about wiring; it's everything in development, everything in um, how bodies are formed involve some coin flip. And I think there are cases where nature can really bias that coin, and we don't perceive it as a coin flip, right? Something in fact, many things that are super important, right? Instead of flipping a coin that's 50-50, you flip a coin that's 99.9% versus 0.1%, right? There's always going to be some stochasticity, but what nature gets to turn is the knob that determines just whether it's 50% or 1% or 0.01% or 99.9%. That's how I think about it.
Right. Something that also pops into mind when we talk about this stochastic nature of development and how our genes specify sort of the architecture and the position, but then there's so much environmental importance in figuring out exactly how those neurons wire up. I've always been interested in those quite tragic cases where, people are deprived of a particular stimulus. How do you think those relate? When there's a lot of cases of the Romanian orphanages when you just had a large amount of Romanian babies basically put into orphanages and deprived of empathy, deprived of any care, loving, light. Like how do you think that impacts that stochastic development of the brain and the wiring?
Yeah. So, the way that we formulate this, nature, nurture, and randomness, it's kind of arbitrary, right? So, I mean, we are humans because our genes build human brains. Our dogs have genes that build dog brains. There's no amount of environmental change is going to turn a dog brain into a human brain or vice versa. That said, our environment will definitely determine aspects of how our brains get wired up. And so, the best studied case, or arguably the best studied case, is it's what actually Hubel and Wiesel won the Nobel Prize for. It's how we develop the binocular vision. So we have two eyes; they each send wires to the brain. And the two views of the world that we get from our two eyes have to sort of mesh up by the time we get to the visual cortex, right? Mm-hmm. And that is how we are able to construct binocular vision, why we're able to get depth perception from our two eyes. The events that allow the two eyes to sort of agree and get their connection to mesh up occur in the first few months after birth. And if you, for example, lose vision in one eye, even temporarily, in animal models, we can cover up an eye of an animal after birth to prevent these connections from forming properly. They don't form properly, right? And this is just, the best studied example of a ubiquitous process, which is that what we call normal development is the result of the right environmental inputs coming at the right time, right? And so, I think you're a native speaker of English. My parents actually spoke Hungarian at home. My parents. So my first language was Hungarian. So we, all babies um, expect, in some sense, for normal wiring to occur. They expect the adults around them to be speaking language. But the details of those languages, right, that of course depends on who your parents are. Now, human babies are pluripotent when it comes to learning languages, right? Yeah, my children um, learned English at home, and their brains were just as good at learning English as they would have been at learning Hungarian. Um, you know, so in that sense, any language is perfectly fine. But the absence of language, going back to the Romanian question, right? The absence of language is pathological, right? And so the brains of those kids, for example, in some cases, actually, I think in the Romanian orphanage, I think they still end up speaking because they got a little bit of language. But there are cases where they don't get any language, and that is sign language, by the way, can also count as a language. The same circuits are invoked, but it's not just hearing. So, but that's part of this larger set of events that humans need to develop along the normal trajectory, which isn't just human interaction for communication for language, but love and empathy, right? There are circuits that won't get wired up properly if humans don't get touch, if they don't get empathy, if they don't get caring, if they don't get if they don't get love, right? And on the one hand, it sounds very reductionist to say there's a circuit involved in love, but I don't think so. I mean, as a scientist, I believe, as a neuroscientist, I believe that everything we are is because of our brain on the one hand, but then I look around and part of what we are is love and compassion. And so then, one of the really interesting questions is, what are the circuits that enable humans, and certainly not just humans, other animals to experience love, compassion, caring, right? And we are beginning to understand what those circuits are. And so if you don't have those as part of the developmental trajectory, if the inputs aren't right, then things can go very awry. And it remains an open question in many cases, how much, if you bring it in later, how much of it can you recover?
Yeah.
And I
There's a case that fascinated me. I made a video on it a while ago called "The Wild Child of Avron." I'm not sure if you've heard of this case of the boy who,
that particular one.
boy who was found in the woods in Avron, France, in the 1800s. So
The most famous one.
yeah, so like 1805, a lot of details, obviously, since you're 200 years back, are uncertain. But he was sort of found in the woods around the age of like 11, 10, 11, 12, suspected to have, what we would now call as very atypical, possibly autism, maybe schizophrenia. And he went through this learning process with a physician, and basically he was able to learn some basic things over time. Apparently he didn't know how to cry or sneeze. So there's a lot of these basic. He had no temperature sensitivity. Again, some of these details we can be a little bit suspect on because we don't know, right? It was 200 years ago. A lot of the documents apparently were destroyed by Lucien Bonaparte, which is a interesting detail. But he never learned to speak. I guess is the conclusion of that. Is that after many years of dedicated learning, the one thing he was never able to do was speak. He learned to empathize. He could learn to hug. He was able to clean things up. He was able to clean himself. He figured out a lot of things, but he was never able to speak. So that's sort of one of those ideas behind the critical language hypothesis, which is like there's a critical period to which you can wire a language, and after that, for some reason, the plasticity in that area just goes away. It's an interesting example. We can, there's a lot of things that we don't know about that's a bit shrouded in mystery, but it's an interesting one to indicate your point, I think.
Yeah. I mean, fortunately, there's precious few of these, but there are some of these cases, and it's just fascinating. What does it? I think one of the challenges, and you sort of hinted at that, is that in a lot of the cases, well, at least in some of the cases, the kids going in may have been atypical in some way. In some cases, it's just outright abuse. I mean, in all cases, it's abuse, but in some cases, there's abuse overlaid on something else, and it can be hard to tease those apart. I think that's one of the. So once again, I'll just sort of circle back to the fact that I study animal models because I just don't believe humans are special. Um, we're mostly very, very, very similar. We think we're very, very special.
Yeah.
But modern humans evolved 100,000 years ago. And there are probably things that are very like us going back about 1 million years, like our common ancestors with the Neanderthal. It's not clear that Neanderthal were any less smart, any less compassionate than we were.
Yeah.
So, but still, 1 million years is a blink in evolutionary time. Animals have been around for over 500 million years, and mammals have been around for over 100 million years. And most of the things we do are already present in animals, if not in animals, then in mammals, and if not in mammals, then in primates. And, the one thing that I think makes us special is language. And I find that really interesting. But, that I think that is what has enabled a bunch of the things that we think are special, like, high level cognition and logic. But, I think those are sort of details compared with the really general principles that apply just as well to humans as to other animals and mammals.
Yeah. I'm really curious on how your work into connectomics has informed your understanding of things like emotion in humans. Emotion is something I'm fascinated in. I think the neuroscience of emotion is shrouded in mystery, but it's, very interesting. How, when you attempt to wire up large parts of rat and mice brains, how does looking at that data and thinking about that data and gaining an intuition about what it means, how does that inform your understanding of something that's quite abstract like emotion?
Yeah, so, emotions, it's sort of funny because it's hard to really define from a sort of in even somewhat rigorous neuroscientific way what is an emotion, what is something else. I mean, there are a couple of layers to that, right? There are, the very I guess the way I tend to think about it is this, that our neural circuits evolved sort of our neural circuits evolved from the circuits of simpler organisms, or at least from organisms that didn't have many of the things that we have. This is just, I guess, we're discouraged from thinking about things as simple or complex, but many of the circuits that we have are evolutionary innovations compared with earlier circuits. But all animals have to solve some really basic problems, like they have to solve the problem of recognizing when they're hungry. They have to solve the problem of recognizing when they're thirsty, recognizing when they're tired, recognizing when they're horny. These basic, you sort of laugh because these are so basic, but like whether you're hungry or thirsty or tired or scared or horny, right? These are going to determine what you do. Some of them are physiological, right? And some of them require more input from the outside world. And these are the basic, sort of problems that even worms have to solve, right? They might be hungry, but they might also be scared because there might be a predator. Well, they have to first figure out what it means for there to be a potential predator there. And they have to decide, am I going to go in this direction where there seems to be food, but there's also danger, right? And as we evolved, we got better and better at sort of solving those problems in more and more complex ways. And at some point, right? So, for example, if you're a simple organism, you go in some direction, you eat some food, and the food turns out to be poison. Okay. well, you probably shouldn't eat that food again. But maybe you don't even want to go in that direction again because that's the direction that has poison food. So you might say, I don't like that direction, or I don't like the color of that food, right? And that's sort of the building block, the first building block of how you get what's eventually going to be emotion. So what we call emotions, I don't know, like, do you want to call hunger an emotion? Probably not. But do you want to call disliking something an emotion? Maybe not. How about feeling unhappy about something? Well, that I think we're getting into something that we might be tempted to call an emotion. We can go on like this, but basically, almost everything we do, almost all our behavior, right, is the result of sensing what's out there in the world and then attaching value to the things that are out there in the world and then making predictions about our future actions. And the emotions sort of come in to that whole process in a complicated way. I should add that if you want someone who's really, I could talk a little bit more about this. But the guy who really wrote this out in just one of the most beautiful books that I've read in neuroscience in like a decade is Max Bennett.
Max.
Max Bennett. Yeah, I highly recommend. His book is called something like The 5 Breakthroughs in Intelligence.
Fascinating. I haven't heard of him.
Yeah, you should read the book and then invite him. He has really sort of laid out this evolutionary approach to thinking about how neural circuits emerge and what role emotion, how to think about emotions in that context.
Makes sense.
So I get, circling back to the question, what we are doing is we're breaking the question of what is an emotion, right, down into the constituent circuit and sort of seeing it as the interplay between all these different drives and sort of I think the right way to think about emotion per se is sort of at the persistent state that is associated with these drives that guides our behavior for an extended period. It's a very dry, in some sense, it sounds like a very dry way of thinking about things, but like it's sort of for me, the fun of being a neuroscientist is this sort of, on the one hand, of course, we are humans and we experience our emotions, but then we're also sort of taking apart this machine and asking, why should we be experiencing emotion? And, we know what it feels like inside to feel this emotion, but what are the physical substrates of this that enable us to feel that? I guess I'll just leave it by saying that even though I've been thinking about these problems since, this is why I became a neuroscientist, is trying to relate what I feel subjectively to what I see objectively, right? To my recognition that this has a physical substrate. Honestly, I still can't, I still don't see the connection in some sense. Like it still hasn't helped me understand. We still haven't answered the fundamental question, which is the one that drove me into neuroscience, which is how you go from 3 pounds of brain to everything we think, feel, experience, how you get consciousness out of that. And for me, that's the fun. It's like one day, hopefully we'll see how to make that connection. But at least as far as I can tell, we haven't yet.
Yeah. Your thoughts and ideas are a lot more complex, but no more conclusive.
Yeah, exactly. We have a lot of data, but we don't have that fundamental, sort of breakthrough of understanding. I'll just say that, like, it's actually Francis Crick, who wrote in a book, I think it was in The Astonishing Hypothesis, that when he started his career, so I guess he was born in probably the late 20s. He said when he started, or at the beginning of the 20th century, he said there were 3 fundamental questions: the nature of matter, the nature of life and the nature of mind. And, by the time he got to college, in I guess the 40s, he felt like, okay, we kind of understand the nature of matter. Sure, physics isn't 100% solved, but we kind of got the big picture there. The nature of life was still an open question. And of course, Watson and Crick, the double helix, he helped answer the nature of life. And we now sort of, again, there are a lot of things in biology that we don't understand, but we kind of have the outline of how you go from molecules and all the way up to cells and beyond, right? We sort of see how to get there. I don't think we've gotten there in that last one, the nature of mind. We know that somehow thinking emerges from the connections of a lot of neurons talking all the time, but we can't make a much stronger statement today than we could when I started graduate school a while back. Like, we still don't have that sort of, we're missing some piece. We're missing some fundamental insight, I think.
How do you think AI might help answer a few of those big questions?
So that's a great question. I mean, I started my career as a computational neuroscientist back when computational neuroscience was the same field as neural networks. They were the same people, the same field. We went to, and I did my PhD sort of at the border between computational neuroscience and artificial neural networks. I should say that when I was in graduate school, AI was distinct from artificial neural networks that usually referred to symbolic AI. But these days, symbolic AI has been largely replaced by artificial neural networks. So you could say that, I did my PhD at the border between computational neuroscience and AI. And the reason was that I felt like if we couldn't build a system that mimicked what the brain does, then we don't understand what the brain does. I see no reason that we shouldn't be able to imitate what the brain does. For a long time, we weren't getting any closer. The algorithms just weren't working. So as I'm sure everyone's aware, at this point, in a lot of ways, we seem to have gotten a lot closer. Certainly when it comes to, the thing that just blows me away is the tremendous progress we've made for language, right? Language in the last couple of years, ChatGPT is just mind-bogglingly good, right? I think for all intents and purposes, it passes the Turing test. It can essentially mimic the output of any human. Yeah. But that said, there's this idea called Moravec's paradox. And what Moravec was a roboticist from the 80s. And he has a great quote, which I recommend to your viewers to look up on Wikipedia. I wish I could quote it from memory because it's really beautifully written. But the core idea is that what computers find easy are things that often humans find very difficult. So, back when when people started building computers, they thought that there would be sort of a one-to-one match between essentially human development and computers. So they figured that, interacting with the world would be easy and chess would be hard. And as we all know, it's the other way around, right? Yeah. The first chess playing computer beat the world champion, I think in 98, Kasparov. Yeah. But, for a long time, you couldn't build a computer that could even recognize the chess pieces, much less manipulate them. We've made progress on the recognition. So when I started graduate school, it was still the case. No computer system could take a picture and recognize the chess pieces or a dog from a cat or a tree. That's no longer the case. Yeah. We now have computer systems that are okay in visual recognition. They're amazing in language, but they still can't do the things that all animals can do. No robot that can, effectively walk around in a complex environment. No, there's no robot squirrel that can climb a tree. Um, so we still can't do the things that are there's no autonomous agent that can engage in like a long extended period of goal directed behavior where the goals are complex and it can balance those. In other words, you don't have artificial systems that come anywhere near to what animals have to do by definition. But you have ones that, they're now, I guess just this week, DeepMind developed a system that could perform at the silver medal level in the high school international math competition. So we're talking, hard math, right? Stuff that, like silver medal of a high school mathematician is hard math. And these artificial systems are now at that level, and probably in next year they'll be gold medalists. So the things that we consider hard and challenging turn out to be easy, but the things that we take for granted, which are interacting with the world in a goal-oriented manner, remain out of reach for modern AI systems. And I think that sort of trying to understand why that is will tell us a lot about how our brains work and what the sort of real challenges are. And that's actually what a lot of my current research is on. It's trying to understand, why are those things hard? Why is vision hard? Because as I say, there are vision systems that in some ways seem to be performing well, but they're not performing it the same way that our systems are. And I think that the differences in how those systems are trained and how those systems actually perform visual computation will tell us a lot about how we do it and how we can build better systems in the future.
Yeah. I'd love to dive into your work. I know you're doing a lot of projects at the intersection of neuroscience and AI. What are some of the big questions that you're tackling?
Yeah, so the field that I'm interested in is called, or by some, it's a new field, but I think we can sort of call it neuro AI. It's the intersection of neuroscience and AI, and it starts from the observation that modern AI, most of the breakthroughs came from looking at how biological systems solve problems, abstracting them away in the right way, and then building systems. So artificial neural networks, which are the workhorse of modern AI, are an abstraction of how neural systems work. One of the big breakthroughs there for vision, as we were talking about, is convolutional neural network. And that's an abstraction of how the visual system is wired up. Jan LeCun looked at how the visual system was wired up, the work of Hubel and Wiesel, and he sort of tweaked it a little, and that's how vision systems work. And we could go on and on. Reinforcement learning is a field that grew out of sort of careful study of how biological systems interact with the world. And I think there's a lot of further inspiration that we can get. And I'll talk about some of the ones that I find most exciting. So the most obvious is that the hardware that we use to run neural networks is incredibly energy inefficient. So training a large language model takes an amount of information, or takes an amount of energy that is enough to power the city of Boston for a week. So it's just mind-boggling.
Wiles, yeah.
It's just wild how much. So it's one of the major costs in these systems. It's just the energy that it takes to run, right? Training a child to speak does not require that much. It requires, a couple years supply of applesauce and cookies.
Right. It's just a phenomenal comparison. It's absolutely mind blowing.
So there are a couple reasons for that, but one of the biggest reasons, so it's a little bit, I mean, although that's the most dramatic comparison, it's a little bit unfair. because in one case we're comparing learning, and the other case it's not exactly clear how you compare learning and runtime. But even on the runtime, our brains take 20 watts of energy to run, and a modern GPU requires, I forget how many, but it's why gaming PCs run hot.
Yeah.
Orders of
magnitude.
Orders of magnitude, depending on which GPU we're talking about. It can be, hundreds, if not thousands of watts per GPU. And that's for, one GPU does not have anything like the capability of brick. So why is that, right? And I think that we kind of know some of the main reasons. The biggest reason is that neurons in the brain communicate through spikes, and there aren't a lot of spikes. So you have 100 billion neurons, maybe 10, 20 billion neurons in your brain. Each of those in your cortex, each of those neurons fires less than one spike per second. And the way to think about that in digital terms is that, you could think about neurons talking to each other through ones and zeros the same way that electronic circuits do. But the cost of a one, the only thing that costs energy is a one. So when you actually send a spike. And so instead of, having ones and zeros that happen about equally often as they do in any digital system, you have a one, if the basic time slice is about 1 millisecond, you have a duty cycle where only about 1 in 1,000 events is a one and the rest are zeros. So that drives the cost of, the energy budget is now like limited by the number of ones you're sending, and only, 0.1% of the time or less you send a one.
Gotcha. That makes sense.
That already, right, will drive down, for a given number of processing elements, that drives down the energy budget by a couple of orders of magnitude. The second thing is, and this is just crazy to think about, so part of the reason that in digital circuits, it takes a lot of energy is that you want to make sure that when you send a one, you're 100% confident that you're sending a one and not a zero. You should never make a mistake. That's why computers don't make mistakes, is that a one is a one, a zero is a zero, and you spend a lot of energy to make sure that you don't mistake the two. If you sort of relax that a little bit and you're a little bit sloppier, you can get away with, so basically that determines the voltage at which computers run, usually like a couple of volts, right? To make sure that ones and zeros are separate. In the brain, in the cortex, you're operating in a regime where one neuron will send a message to another neuron, but because of stochasticity, 50% of the time that message might not go to the target. It just fails across the synapse. In fact, at some synapses, it can be 90% failure. So you can be really sloppy. The cortex somehow operates in this super sloppy, noisy regime, and yet it works. And so we don't know how to build a circuit that operates in a regime where you only send a signal, a spike, 1 in 1,000, 1 in 10,000 time slots, right, where you can throw away 90% of the information in some sense, and it still works. Yeah. So one of the sort of really, to my mind, exciting potential contributions of neuro AI is figuring out how you can compute in the regime that we know that actual cortical circuits compute. If we could figure that out, we could build hardware potentially that could exploit that and drive down the cost, the energy impact of computing by orders of magnitude. Right. So I think that's one of the huge potential contributions, but there are others too, right?
That's a really interesting example. I just recently had Mark Humphreys on the podcast. He wrote a book called The Spike, a brilliant communicator. But he explained this to me a while back that I didn't really know, is that neurons and spiking follow this extreme Pareto distribution, where like 15% of neurons result in like 90% of the spikes. You know, I somehow went through an entire neuroscience degree without figuring that out, or maybe I was just not listening that day. But that was pretty shocking to me. You know, that to me that almost seems like wasteful energy in that they're firing spikes and it's not resulting in any, you know, physiological mechanism. But the way you say it makes sense is that it's incredibly low amount of energy, and that the energy constant, it's actually saving energy by doing that. And the fact that computers can't mimic, or we haven't figured out how to do that in a computer system yet, is why it's so energy efficient.
Yeah, so I think it's not surprising that you made it through a neuroscience course without knowing that, because people know these facts, but since we don't know how to compute in that regime, we kind of push them aside and then operate in, our models operate in this very different regime. Um, and so, you could have blinked. Maybe not your neuroscience instructor threw that little fact on about the sparseness and long tail distribution of spike, but because we don't know what to do with it, like if you actually look at the models that most people are using for computation, they're not exploiting that fact. They're assuming that all neurons interact, all neurons are firing at about the same rate, for a pretty high rate, that all that matters is average spike rate, et cetera, et cetera. And we just know that that's not what's really going on. But since we don't know how to make that work from a computational point of view, how to exploit it, it just sort of gets glossed over. So I think that's one really exciting area.
Is that kind of what I learned about brain organoids a couple of months ago, which are also a widely fascinating thing. Do you think that's sort of the question that brain organoids are trying to solve? Maybe give a quick sort of overview on what they are and kind of what they're doing.
Organoids are super interesting. They're basically a technology for. So we've known for a long time how to get neurons to grow in a dish. But if you just grow neurons in a dish, usually, like traditionally, it's in a flat dish, and neurons sort of wire up. And you can learn a lot about synaptic transmission, but you can't learn a lot about neural circuits, because the way they wire up in a dish traditionally has been, just so completely different from how they wire up in an actual brain. So what organoid, so for the last, I don't know, 10, 15 years, there have been better and better technologies for growing things that are like organs, hence the term organoid. There's been progress in making liver organoids and heart organoids, and sort of complex tissue that's in a dish that has some of the structure that it would have had if it were in the organism. And so brain organoids, right, are the attempt to make sort of a bunch of neurons grow together in a way that's reminiscent of what they would have been like if they were growing inside a skull. And so what can we learn from those? I think one of the main things that we learn is just, again, going back to these earlier questions about development. What are the rules that allow collections of neurons to wire up in the right way? We can also ask, if you kind of have them grow together in the right way, how do they behave? I'm not, at this point, if I'm studying a computation, I still sort of am inclined to look less at the organoid and more at the activity of neurons when they're inside a brain. Right. Because there you can be really, I think it's, sort of crucial to understand neural activity in the context of behavior. And even though organoids, you get patterns of neural activity that are reminiscent of the patterns of neural activity that you see in the intact animal, they're not driving behavior because those neurons aren't connected to a body.
Right, right. What are the main differences between biological neural networks and artificial neural networks? What are the crucial differences?
So this is the question of neural AI. I mean, I can fill an entire textbook with what the differences are between brains and artificial neural networks. But what you asked was not what are the differences, but what are the key differences? And the problem is we can only work backwards, right? What we can do is we can take inspiration from what we know is actually there, and then we try to put it into an artificial system. And if it helps, then we say, that was a key difference. So I can tell you, but this is aesthetic. This is opinion, right? So if you ask me, I just listed one of the key differences, or two of them actually, which is that that neural circuits communicate through spikes, and the spikes are sparse and they're stochastic. So spikes and stochasticity, those are the two of the key differences. But I'll tell you a couple others that I think are really important, which is that I think the key difference, perhaps even more fundamental than that, is going back. This is why I study neural circuits, is that the function of a neural network is essentially 100% encapsulated in the wiring diagram. If you use an algorithm to train up a neural network, what that means is you're essentially determining what the connection diagram is between the neurons in this artificial neural network. The wiring diagrams of brains are a tiny, tiny, tiny subset of all possible wiring diagrams. For example, neural systems, each neuron tends to be connected primarily to local neurons because sending an axon a long distance is expensive. And so mostly you want to keep your axons local. Recently, this is why I've become interested in development. One way to think about it is that the set of wiring diagrams that are possible are those for which you can write down simple developmental rules. And so what we've been working on is sort of mathematically characterizing the kinds of rules that nature can actually generate. And that greatly constrains the kinds of wiring diagrams that can emerge. And by looking at those kinds of wiring diagrams, it potentially provides insight into how these computations work. So one thing that I think turns out to be really important is that the kinds of wiring diagrams that we can build are essentially those that can be built by recursive rules, right? You connect. You basically keep reusing the same rules to build wider, bigger and bigger modules. Anyway, these are some of the things we're thinking about. We're, like I say, the history of AI is the history of squinting at the biology and then asking, how can we take that idea and convert it into something that, when we put our engineering hats on, we can actually use.
Right.
Yeah.
Where do you think the current approach of scaling data sets and compute will get us in terms of near human cognition, consciousness, sentience, AGI, sort of whatever label you want to tag on it? What's your sort of intuition there? Assuming hypothetically we're not limited in data or compute, we just hypothetically we have infinites of both.
I guess that changes the answer. The fact is we are limited in data and compute. So, I mean, there is a, sort of mathematically trivial sense in which if you had infinite data and infinite compute, you could just list all possible, you could generate behavior by a lookup table, but that's not very aggravating.
Yeah.
Right. So, look, I was shocked at how incredibly impressive GPT-4 was when it came out. I mean, I was making strong predictions that you couldn't get anything like that. I had all sorts of strong beliefs about how in order to really get language to work, you'd need grounding. And it turns out that we've learned, to my mind, GPT-4 and Claude have taught us something about sort of the degree to which language is a self-contained system. But, GPT-4 was released over a year ago, and we have now seen a year of sort of incremental improvements over the GPT-4 that was released a year ago. There have been a bunch of Claude, I think, is just as good. I don't think Gemini is quite as good, but I think there are tricks of the trade. But what we haven't seen in the last year is the promised GPT-5. Will we get a GPT-5 that's a quantum leap over GPT-4? It's an open question. Given how wrong I was about GPT-4, I'm reluctant to make a strong prediction, but I'll say I'm skeptical. Similarly with vision, although, it's incredibly impressive what vision systems can do, they're still sort of not doing it the way we do it. And I think that means that they're ultimately going to keep failing on edge cases, right? Like if you think about the most commercially important application of sort of vision decision systems, it's self-driving cars, where there's been, I've heard estimates of over $100 billion of R&D because it's such a, Elon Musk promised in, I think, 2016 that by 2020 we'd have fleet of self-driving cars, and there was a lot of hand-wringing about all the drivers who are going to lose their job. We're now in 2024, and, self-driving cars are kind of okay, but I got to say, when I've been behind the wheel of a self-driving Tesla, on the one hand, it's impressive, but on the other hand, it's more exhausting than driving myself because I'm so terrified of the rare. But I think this is important, right? Because what we're getting is this, where they fail is this long tail of edge cases, right? And when you try to solve these problems by brute force data, you're always limited by the fact that there's, this long tail of increasingly unlikely problems that can come back to bite you. And they don't, and what that's telling you is they're not solving the problem the same way we are, right? And so what I think, going back to the question of what NeuroAI can answer, is that the answer isn't more data, right? There's this great example that I sometimes do in my talk where it's a couple years old, but it's easier to understand than, what it means to have a long tail. Vision system from a couple years ago, we get fooled. There were these two pictures of a cow on a grassy background, which the system easily identified as a cow, and a cow on a sandy background, the kind of skinny cow, which the vision system calls a camel. Right. And why? Because, so on the one hand, you could say, well, we just need to show it more pictures of cows on sandy backgrounds and understand the cow on a sandy background. But that's not really the problem. We're never going to find enough images, I would say. That's why I'm saying that, like, this idea of scaling, for every tenfold more data, you get a, you drop your error rate by maybe another factor of 10, but you need a lot of factors of 10 before you're happy, especially with things that matter, like self-driving car, right? And these are mistakes that we would never make, and that honestly animals don't make, because more fundamentally, we know not to give that much attention to the background, because we know that animals move with respect to their background. And so there's a fundamental problem that I think isn't going to be, and this isn't going to be solved just by more data. It's going to be solved by building systems that not only solve the problems we do, but solve them in the same way that we do.
Right.
And the way to get them to solve them the same way is that their foundation should be similar to the foundation from which the foundation that we humans had as we evolved from, things that were like rodents to primates to, apes to humans. And I think sort of following a sort of trajectory like that, an evolutionary curriculum is going to be key. That's where I'm placing my bet. Is the idea that we could train these systems potentially with orders of magnitude less data if their foundations were the right foundation.
Right.
And that's what I think is the interesting problem.
That's a really good example, I think, with self-driving and how it's fallen very short of predictions. Would those edge cases that you talk about in the vision systems, would an optical illusion be a good example of an edge case for humans, where we're looking at it and for some reason our vision system just cannot figure it out, like Escher's staircase or something where just the visual scene doesn't make sense?
Yes and no. So, like, it is an inevitable. So, vision solves this problem of inferring. One of several problems that have to solve is the problem of inferring the structure of a three-dimensional world from two-dimensional images.
2 dimensions, yeah.
And that is, by definition, by construction, a problem where you will occasionally make mistakes.
Yeah.
And optical illusions tap into that, and they tell us sort of the algorithm by which we're solving that problem by figuring out edge cases that are the edge cases that are sort of indicative of the algorithms that we use. So to my mind, the key insight is that the things that are edge cases for humans are not necessarily the same things that are edge cases for artificial systems, and vice versa, right? In vision, there are these so-called adversarial examples where you have two images that are almost identical. They look identical to a human. One is like, you sprinkle a little bit of pixel noise onto an image that looks like a cat, and you can convert that image from, or you can convince the system to relabel that cat as a pig or something. And you're like, what? These two images are indistinguishable to us. Why are you suddenly saying that that's a pig? And that reflects the fact that it's not solving the problem. These are all symptoms of the fact that these systems are not solving the problem the way we're solving them. And I think that's the core problem that we have to address if we're going to sort of make advances. I think that, that's one of the big insights that I would say that neuroscience can provide is not just, that we should solve these problems. We should define these, image recognition problems, but we should try to train these systems so they solve them the same way we do. And therefore, they'll make the same mistakes that we do. And if they make the same mistakes, then they can honestly sort of be incorporated into human environment, right? We won't be satisfied with a self-driving car that makes fewer mistakes than human. Yeah. We want them to make not just fewer mistakes, but the same kinds of mistakes. Because, I'm going to say that even if you could show that a self-driving car had 10 times fewer mistakes than a human driver, if one of those mistakes is running down a kid playing in the middle of the street under conditions where a human would never make that mistake, I don't want that self-driving car, and I don't think anyone else will. Of course. So it's got to make, if it hit that kid because the kid ran in front of the car, and you look at the video, it's like, yeah, there's nothing that could have been done. It's sad, but it is what it is, right? Yeah. All systems will make mistakes, but if it just runs that kid over, I don't want that in my environment, and I don't think anyone else does. And so I think we're not going to accept sort of machines in our environment until they behave sort of the same way we do. And I think that means sort of going back to in some cases, sort of reimagining how these systems are trained.
Yeah. So how can we do that? How can we take the lessons of genetics and evolution and start a system off in a really basic, primitive state, bring it through millions of years of trials, hypothetically in a few months of training, and then arrive at a foundational model which has whatever the correct ground truths are, right? Because these models are being called foundational models, GPT-4, and people are fine-tuning them for all these different use cases. I ran a startup for 4 years where we were doing a very specific type of translation with these foundation models. Like, how do we get to these foundation models that have the right ground truths? I agree.
You just said it, right? Like that is, you just outlined what I think is the right approach. The details of how to do it, right? Like, I think that that's going to require a huge effort. I'm excited about participating in some of these, but yeah, I think we need to go back to square one. I'm at this point, a fan of these simulated environments. I think what we need to do is training large numbers of agents in simulated environments, have them interact, and then sort of have them evolve. I'll just say that, as a technical matter, I'm not a fan of evolutionary algorithms. I think one of the main things we've learned from machine learning is that if you can find a way to tweak parameters through with a gradient, that there's a big win there. So even though I like the process of evolution, I think we can speed up evolution by orders of magnitude through a gradient. So what I envision is setting up artificial environments of interacting agents. The environments, I think one of the challenges, it shifts the burden, because now the challenge is the environment has to be diverse enough to challenge these agents to do interesting things. Yeah. I think that's going to be one of the main challenges. How to do that? I've been thinking a lot about that, talking to people about that. There are some existing efforts, and I think those are worth doing. I think people sometimes get disappointed. I'll just make a side point here that people sometimes get disappointed with simulated environments for robotics, because they find that the transition of so-called sim to real is very difficult. So if you try to train like a robot arm in a simulated environment, and then you move it into the real environment, it doesn't carry over perfectly.
Right.
But I think that tells us that what we need to be getting in the simulated environment is not every detail, and then expect that agent to interact with the real world perfectly. What we need is to train these agents so that they're flexible enough to interact with the world. And one example that I think about a lot is that, if you train a robotic quadruped in a simulated environment to walk, what you get, you can train a simulated quadruped, but the so-called policy that you get, the agent that you get, if you change that quadruped dimension a little bit, it won't work anymore. And that tells you why it doesn't work when you bring it to the real world, because the policy that you've gotten, what you've trained, is typically pretty inflexible. By contrast, right? Think about dogs, right? You can mate a Chihuahua with a Great Dane, and you get a puppy that has, Chihuahua brain, Great Dane brain. Yeah, and you get something that works. So the same neural circuit, right, works between a Chihuahua and a Great Dane and everything in between.
Right.
And within, a couple weeks after birth, the thing works, right? And if you think about horses can stand and run within minutes of birth. So whatever neural circuits they are born with innately are able to control a complicated body in a way that's infinitely more flexible than anything that we know how to train artificial agents. And that is the kind of thing that through this evolutionary curriculum, I think we ought to be able to get to. That's, I think, the potential pair. We know that biology has solved this problem. Yeah. In some sense, it has to deal with the sim to real problem, but it solves it way more effectively, right? Right. In some sense, the brain that it's born with has only ever dealt with all possible environments, but then that brain is stuck in an actual body. And in most, humans are always the worst. Humans take a couple of years to learn how to walk, a year or two, but that's not the right thing to think about, right? It's animals that can walk minutes after birth, or in some cases, if you're an insect, after.
Yeah.
Right. So I think understanding how neural circuits can sort of pop into the world ready to form, ready to engage not just in, I'm talking about locomotion because it's an easy thing to study and think about, but everything, right? We enter into the world, animals enter into the world ready to go. For the most part. Yeah, we get a little extra information from the environment. We do a little learning, but we're mostly ready to go. And we really don't know how to do that yet. And I think that's what, if we were to follow the right evolutionary curriculum, and then each generation have that agent sort of follow a course of development in the formation of its neural circuit, that's where I'm putting my money. That's sort of the vision that my research program is following. But who knows, right? Maybe brute force from AI and engineering is going to get to it. I don't think so.
Well, it's such a cool idea. I think it's a cool idea because it's so human. Because like every human went through this process, right? We went through the process. I mean, mostly subconsciously for a long time, but then consciously of just being dropped in the world and getting stuff thrown at us and was figuring it out. So this idea of building AI agents in the same way, I think, is very human. It's a very innately primitive. And then that also means that when it makes mistakes, we can sort of understand why, or we can even empathize with it possibly, because we may have also made that same mistake in the same environmental constraints. I think one of the difficulties with the AI systems at the moment is that, one, we don't know what they're fundamentally doing, right? I mean, one of these big issues is in the language models is that there's just a mess of computation, but there's like a big question mark on what actually is happening there. We don't know why it hallucinates. Hallucination is a huge issue and we can't figure out the hallucination problem. But as long as we just don't fundamentally understand the mechanism, we aren't really going to be able to make much progress with it when it doesn't work. And in this way, at least if we have a very flexible system, we can understand the errors that it's making and then be able to correct it on the fly and correct it on course.
Yeah, potentially. I mean, I think that our inability to understand, in some sense, what these large language models are doing, it gets at the question of what understanding would be, right? Because, as an experimental neuroscientist, right, our dream would be to be able to do what we can do in large language models. I spent a lot of my career thinking, if only I could sort of know what the neural circuit is, that I could understand what's going on. Well, in these large language models, we can know absolutely anything we want to know. We can make any measurement we want to make. We can observe the activity of every neuron. We know the value of every connection. And yet, it's very hard to get at what we would call understanding, as you said. I don't know if you saw this blog post a couple months ago on Golden Gate Claude.
No.
Oh, okay. So the Claude language model from Anthropic, right? They did some work on what actually is going on inside of it, and they used some cool techniques, variational autoencoders, to figure out sort of the subsets of units that were simultaneously active, and then they found sort of they were able to make, so they found subsets of units that represented sort of high level concepts like anger or quiescence and then various words. And then to show that these subsets of units were actually causal, they would crank up the activity on them. And I think there was like angry Claude that was always, instead of giving the normally polite responses that these large language models make, it gave rude and angry ones.
Really fascinating. Wow, that's so funny.
Yeah, exactly. And then they released one of these models called Golden Gate Claude. For a while, they had an interface you could play with it. So they found a subset of neurons that they thought represented the Golden Gate Bridge, and then they just by hand tweaked the activity of the subset of neurons, and then this large language model, this particular instance of it, was obsessed by the Golden Gate Bridge. Every answer would somehow get directly back to the Golden Gate Bridge. And so what are the causes of World War II? And say, well, World War II occurred as a result of the meetings of the Allied powers in San Francisco in the 1930s within view of the Golden Gate Bridge. And
Brilliant.
and I think there was one where I think it was explicitly asked not to mention the Golden Gate Bridge, and it would sort of produce a sentence or two, and then it would mention the Golden Gate Bridge, and then it would apologize. Oh, I'm talking about the Golden Gate Bridge. I shouldn't be talking about it. Oh, let me see if I can fix that. And then it would do another sentence, and it would apologize again. And it was just like oh, this body had OCD. It was having intrusive thoughts about the Golden Gate Bridge. You can look it up. I think there are. If you Google Golden Gate Bridge Claude, it's hilarious.
I'll need to look that up. That is hilarious. Thank you so much for bringing up that example. I can't believe I didn't see that. That is so fun.
Well, yeah, it's a lot of fun, but it also, it gets to this larger. I think they have some academic papers that are, I don't know where they, but it's a serious piece of work, and it's really sort of informative for thinking about like what it means to understand a system where you can you can see the activity of every neuron, and still you don't know what to do with it.
Yeah.
Right. And the worst case scenario is that that's what our brain is like. I think what you're alluding to is the hope, and I still share that hope, that because our brains evolved, that maybe there are sort of guiding principles that will allow us to sort of understand the activity in a more satisfying way than I think the way we're understanding Claude. Because on the one hand, we do understand something there, but I don't know. I hope that we'll have some kind of understanding that's better than,
Right.
more satisfying than Golden Gate Claude.
Yeah. So what, in an ideal world, what collaboration needs to happen between neuroscience and AI in a perfect scenario where you have research effort, the best of research efforts on both sides of the aisle, that is merging together to create the best unification, say, in both fields?
So I've actually here at Cold Spring Harbor been booting up a program to bring in people with a background in AI and train them up in neuroscience. So what you need, when I was in graduate school, I was part of a cohort of people who were, if you like, bilingual in neuroscience and AI. And these days, each of those fields is sort of to some extent, gone its own way. Certainly, most AI people know almost nothing about neuroscience. That was not the case, when I was in graduate school.
Right.
People were constantly looking to neuroscience for inspiration. But, understandably, AI has become so mature, and it's such a fast-moving and complex field, and it's so deeply rooted in just pure engineering that you can get pretty far by just, you can certainly make progress just by treating it as a standalone engineering discipline.
Yeah.
But I still believe that the fundamental progress, will come by taking insights from neuroscience and applying them to AI. And I believe that to make that happen, what you need are another generation of people who are bilingual in those fields. And so the way that we're doing that is we're taking people with a background in AI and embedding them in a community of people who are interested in AI but are fundamentally neuroscientists.
Right.
And we've got a small program here, but I think that could be replicated at a lot of different places. So I think that that's really what we need more of, is people who sort of take, not just read the occasional neuroscience paper, but really understand that, as you said, you didn't know that there was a Pareto distribution, right?
Yeah.
Well, like, that's not something that you're going to see in a survey course, right? And yet that could be the crucial fact that allows us to, that inspires us to figure out how to compute in this sparse regime.
Yeah.
And, I'm coming at you with the examples that I've thought of, but there's going to be some, there are lots of ideas that I don't consider, like maybe glial computation. I personally don't I'm not going to place my bet there, but that's not to say that somebody else can't say, oh, suddenly I see how we can take what glial cells do and abstract that in the right way and build a better system, right?
Yeah.
I
Yeah.
don't see how to do that, but maybe someone else does.
Yeah, I love this. I was always very upset that we never, in our entire neuroscience course, we didn't write one line of code. Actually, we did one lab in OR to change the color of the Y and X axes, and that was it. And that was very frustrating because we'd spent about 1,000 hours pipetting liquids from one beaker into another. Obviously, that's required and essential in neuroscience as well. But the fact that we didn't learn the basics of Python to be able to understand computational neuroscience, even on the smallest scale, I thought was upsetting. I would love to see more emergence of neuroscience, computer science, and AI going forward. I'd love to see courses in that. I think would be. I would love to. I'd sign up for that immediately. That
Yeah,
piques
yeah.
all of my interest. It sounds incredibly interesting. I am. This has been such a pleasure. I have learned so much, and I could go all day, but I don't want to steal all of your Sunday. So I'll leave with one final question that I like to ask all the guests. So within the context of neuroscience and AI, for the next decade, what is the one thing that you're most excited about? And then the inverse of that is, what are you most scared of?
Um, well, what I'm most excited about, I, I think kind of by definition, you work on what you're most excited about. I wouldn't be working on it if that's not what I was. So I'm excited about this sort of the new potentials for these uh, for the contribution of neuroscience to AI. The particular versions of it that I'm excited about are the ones that we sort of touched on: the evolutionary curriculum, how to compute with spikes. Um, as part of my day job on the Connectomics, like how can we get better insights into how neural circuits work by understanding those circuits better, and how we can get better insights into the pathological processes by seeing how those wiring diagrams can go awry. Um, Honestly, on the, what am I scared of? I'm not an AI doomer.
Okay.
I'm not worried about scary AI systems that will, take over the world. I do see some pretty scary scenarios for economic disruption as
Right.
AI gets better. And I find that a sort of very concerning issue. I think that I guess the economist John Maynard Keynes predicted in the 1930s that by the mid 90s, productivity would have increased to the point where people would only have to work, I think he predicted, 10 hours a week. And the fact is, it did um, improve to that point where if we had organized society differently, we could have 10 hours a week of work and the rest leisure time.
Right.
But
We just took on more
because
work.
of the way we organize the societies, that's not what we got,
Yeah. Yeah.
right? People are not working 10 hours a week. I think AI is the same, where, it could be incredibly beneficial, but in practice, the way we've organized our societies, at least right now, most of those gains are going to go to the people who, own the companies, to the largest companies. So I see that as very concerning. And then I guess for the progress of science, um, the funding climate in the United States and potentially elsewhere, I see clouds on the horizon. I think a lot of people have, for really unfortunate political reasons, have become increasingly skeptical of science and what
Yeah.
it can contribute. And depending on how, in the short run, how certain elections go, we have an election coming up, that could end up being very bad for American science. But I think that, and even in the medium term, there is an increasing skepticism of science. And I think we scientists have not done a good enough job sort of communicating how science works and how it can be beneficial.
Right.
I think having people understand that when we cure a disease with science, that's the end of a very long road. And at the beginning of that road was sort of fundamental science, where we just sort of were driven by curiosity. How do things work? And you can't just go straight for the disease cures.
Yeah.
And I think just even aside from, the politics of it, it's just even, well-meaning people think, why are we wasting our money on what to them seems like studying flies? Why are we studying worms or flies?
Yeah.
And trying to make it clear that the insights that we get from studying yeast or worms or flies or mice, right, and studying weird questions like how do brains wire up, ultimately answers to those questions pay off 1,000-fold in terms of curing or treating neurodegenerative disorders, neuropsychiatric disorders, diseases like depression.
Right.
Yeah, and I think that like our failure to communicate that in a way that really reaches the general public could have real consequences going forward in terms of funding.
Right.
Anyway,
that was a well-meaning point. I think that the problem isn't. I think some of the problem is in the peer-reviewed journal system, in my opinion, because I think that those things are just impossible to digest and read for anyone that isn't a scientist. So that kind of creates an imaginary barrier between scientists and general population. But I also think that that's the role of specialized science communicators. Scientists are dealing with the hardest questions in the universe, and they should be focused entirely on that. But I think that we should have far more scientific communicators where their entire job is taking that complex information and figuring out how to feed it to the general audience in a way that's engaging, in a way that's enticing, in a way that gets them excited about science. That's sort of what I'm trying to do with this podcast. That's what I'm passionate about doing. Yeah.
I think that that's so important. Trying to reach people like I'm not an expert in anything but a little corner of my science, but I like to learn about other stuff. But it's very hard to learn about other stuff both within science and anything else that I'm interested in.
Right.
Except through these days I rely a lot on this podcast, honestly.
Right.
So I think that's I think what you're doing is just super valuable and important, and I agree. We need 10 times more of them, 100 times more of them.
Well, if I can give, even the smallest mouthpiece to incredible researchers like yourself that is doing just mind-bogglingly insanely cool work. Like it's the joy and pleasure of my life that I get to do this for a living, where I get to read about this incredible science, figure out what interesting stories I can tell in it, and then figure out how to give that to a general audience in story form. That is just the most fun ever. And I think I'm going to commit a large amount of my life to it because I think that there's purpose in it. I think it's very fun.
It's a great calling, and I think you're leading the way. We need more people like you.
Thank you so much, Tony. It's been an absolute pleasure. I've enjoyed this conversation so much. Thank you for giving up so much of your time. I wish you all the best with your future research. I'll have to have you on again because my questions sheet is only half. It's only half answered. I could have gone for so much longer because you're such an interesting guy.
Well, it's been a lot of fun. Thanks so much.
Thank you so much. Absolute pleasure. I'll stop it now.